right. Hello, everyone. We're back here at 11:15 and ready for our third and final session on WeedPedia training. And so we'd like to welcome Koen back. And thank you again, Koen. I know it's getting into your evening hours now there, so we really appreciate the time that you're taking to do this training for us. And look forward to moving into this section on data formats and help people think about how they might upload their own data sets into the WeedPedia for data analysis. So I'll turn it over to you.
Thank you. To start off with the easiest part, a regular user without administration rights cannot upload data to Propedia. And that's mainly to prevent data overload and a simple mess. So only an administrator can upload data into Propedia and assign the proper access for everyone. So of course, as soon as you identify relevant information that needs to be in the WeedPedia, you need to go via an administrator. So this is what that administrator access point looks like. So on the left side, you can simply see a button, Add Upload Files. An administrator has to upload these files, wait for the Propedia backend to process these files. You will, the administrator will see that at the top bar, and then see if no errors are found in the processing. Because as soon as a data format deviates from the standards that Propedia expects, it will simply be impossible to add that to the system. So we have a variety of data formats that we accept, and hopefully the most of them are somewhat familiar to you all. And we will go through a couple of them in extensive detail. So you see here the list of file types that are supported by Propedia. And well, they are almost ordered by popularity, but the most used are obviously GFF, FASTAs, VCFs, BEM files.
Hey, Koen. Sorry to interrupt.
Sure.
I just want to check. It appears that we don't have your slides shared right now. Can you check on that?
Oh, that's interesting.
See if the screen share is working.
Okay, tried it again. Do you see my screen now?
Not yet, no. There we go.
Yeah.
Yep. Okay. Yeah. Now we see the PowerPoint file is open.
Okay. Well, I'm not sure what happened there, but we'll just start again.
Great.
Briefly.
Okay,
okay. So, quick summary again. This is the interface for the administrator. So there's an upload button and a bar at the top that simply states how the uploading goes. And this page is not accessible for regular users. Then finally, here's that list that I was talking about. So Propedia supports 15 data types. And the most popular ones I will go into in detail. So if you're not familiar with these data types, and if you want to learn, then this is a great talk. If you're really familiar with these data types, then it's a bit boring. But otherwise, hopefully it will be beneficial. So FASTA files are one of the most common files that you want to put into Propedia. And these are often the reference sequences that you want to add. However, they can also be genetic sequences or protein sequences or any other sequence features really. And a FASTA file is bound to certain criteria in order to be a FASTA file. So a sequence should always contain a header, and a header always should start with this particular symbol, and then a name behind that symbol. For example, sequence 1, sequence 2, chromosome 1, chromosome 2. And the sequence can only contain ATCG and any IUPAC combination, which basically describes a possibility of different DNA bases. Any FASTA file that does not follow these rules will not be accepted into KeyPedia. So a couple of examples, hopefully obvious one. So in the first entry that we see, there's a SNP denominated by brackets. This is a very common way of stating SNPs in sequences. However, this cannot be accepted by KeyPedia. So if you want to really add this particular sequence to the system, or you have a file that contains many of these brackets, then you need to convert them to IUPAC, because IUPAC is simply a single letter that is accepted. So the second example is, of course, also not a valid FASTA sequence because there's no sequence available at all. The third entry is acceptable. However, keep in mind that using spaces in headers is acceptable. However, that will be only by KeyPedia seen as a description. So in this case, the name of the sequence will only be my_valid_sequence. In general, I highly recommend to not use any spaces in FASTA sequences whatsoever, because many tools simply remove everything after a space. And then you will end up having issues in all sides, in all kinds of other downstream processes. Another common way of writing FASTA headers, and this is also often seen in data from NCBI, is using the pipe symbol or sometimes slash symbols. However, many tools cannot handle these symbols, and also not KeyPedia. So as a rule of thumb, simply use A to Z in the headers of a sequence, whether it's in lowercase or uppercase. And you can have a couple of symbols that are acceptable, for example, the underscore. So that's a FASTA file. Then we go to GFF3, which is the latest version of a GFF format. It stands for a general file format. However, there's nothing general about it. These files are often used to store features with a certain position on a FASTA file. So for example, genes, markers, ESTs, QTLs, you name it, everything that's position bound is often stored in GFF3. And also genome annotations are almost always stored in GFF3 format. A single feature can end up at multiple positions, and especially if you map features to a FASTA file, there's obviously no guarantee that these positions are correct, so always validate your findings there. And a GFF file is much more complicated than a FASTA file. So a GFF file is a column-based file, where the first column stands for the sequence identifier, and that sequence identifier should match to the sequence identifier in a FASTA file. Otherwise, a GFF file cannot be linked to a FASTA file. So if your wheat genome contains chromosome 1 to chromosome 12. For example, I don't know how many chromosomes in general a wheat has, but then you also need to use the same nomenclature in your GFF file. The second column is the source. So where did the data originate from? So that's a free text entry. The third column is the type, and there are various data types defined. And depending on what you enter as a type, genome browser is able to display certain aspects. So keep that in mind. If you want to know more about these types and what's possible, then just have a look at the extensive documentation online. Every single feature in the GFF file needs a start coordinate and an end coordinate. So that's in the fourth and fifth column. And then for some of you, this may be relevant, but a GFF file is so-called one-based. So a position in a genome starts with 1, whereas some other files start with 0. So keep that in mind, that sometimes there is a one-base difference between coordinates. But in a GFF file, it always starts with 1. The sixth column is a score that you may give to certain features. The seventh column is the strands, so whether it's located on the plus or the minus. And then we have the next column to be the phase, and the phase only refers to genetic data types, because sometimes intron-exon boundaries are not very nicely divided by 3. So you sometimes have some leftover content. And with the phase column, you can define how many bases of a particular intron/exon boundary needs to be calculated with the next intron/exon. Complicated story. If you need more background on that, just use Google. Most genome annotation pipelines which generates these GFF files have the phase included. Finally, attributes. This column basically tells how the data is related to each other. So in this case, in the form of genetic content, the gene ID is Eden. The mRNA is Eden.1. And for all the other features that match to this particular mRNA, you simply state that the parent is Eden.1. So genome browsers will know that this set of features belong together. And there is a free text field available in the GFF file, which is the note. And in the note, you can basically type anything. So it's often used for annotation. And this information is then also displayed underneath the data property in the visualization. So it's a complicated file format, and if anything is incorrect, a GFF file cannot be uploaded to KeyGene, and it's quite a tedious process to figure out what may be wrong. And various publicly available databases have different styles of outputting GFF files, which sometimes cannot automatically be accepted by KeyGene. This is the way a GFF file will look in KeyGene. So you have your a genome on top, and these mapped features that we also saw when playing around with the gene passport, for example. That's the visualization of a GFF file. You can also use a GFF file for alignments. So if you have, or if you want to make a multiple sequence alignment, if you have read data in aligned style, then you can also display that in KeyGene. Although the preferred format for that would be BAM files, but I'll come to that. Examples of tools that generate these kinds of outputs is BLAST, GMAP, and Decipher is a hardware accelerated version of BLAST. So validity of GFF files is checked by KeyGene, and apparently there are a lot of common mistakes that you could easily see, but the most difficult ones are obviously the ones that that are not that easily detectable. So a GFF file should always have 9 columns. The start coordinate should always be bigger than the stop coordinate, also on the reverse strands. Attributes are invalid combinations of keys. So for example, in the gene mRNA intron exon structure, you need a proper key value combination, and all attributes should have a unique key, obviously. Then we end up with the gene passports. So these are files that cannot be obtained from the public domain. It's a KeyGene specific output. However, you can also generate it yourself. The only thing that you need is extensive knowledge of JSON. And JSON is a format that you see here. So KeyGene expects a couple of keys, for example, cytoplasmic, blast site ID, and then a certain amount of values. And if you are able to generate your own JSON in this style, then you don't need gene passports delivered through the KeyGene pipeline, even though they are obviously very good. But you then are also able to make them yourself. And these gene passports contain everything that we previously saw when looking at the genes: UniProt IDs, blast results, localization, protein clustering, and Go terms, etc. Making a single mistake in a JSON is incredibly difficult to detect, because JSONs can, in programming languages, nest. So then you have multiple brackets within brackets within brackets, etc., etc. So that's very difficult to spot. So this is a very tricky format to understand by eye. Another common formats, SUM files, so sequence alignment / map formats. These are human readable files. So you see them at the bottom. And these files are often generated when you map sequencing data to a reference genome. A BAM file is the binary format, which only consists of 0s and 1, and that's better readable for a computer than the human readable formats. We also have WIG and BigWig files. Those can be derived from BAM files, and they are often used to display coverage. So collapsing read information to coverage information. And BAM files, BigWig files are supported by the genome browser. SUM files and WIG files are not. And also BAM files and BigWig files take way less storage capacity than their human readable counterparts. These files are often okay, as they are generated by tools that are widely used, such as PWA, Bowtie, to name a few. Then we also have variation data, and all variation data is typically stored in a VCF, which stands for variant call format. And these just contain SNPs, indels, and most recently also structural variation. VCF files are supported by KeyGene. However, VCF files have a certain degree of freedom. That's how I like to call this. So that means that typically a VCF file is not accepted by KeyGene because there are certain fields that KeyGene expects which are not required by the specifications of this format. So uploading VCF files to KeyGene is a sometimes tricky task. But in the help section of KeyGene, you can exactly see how this format should look like. But the most widely used tool to generate VCF files is the GATK toolkit. And these files are always properly formatted, so it's not a problem for KeyGene at all. As an additional thing, KeyGene and JBrowse/Genome Browser only support VCF files in BG-zipped format, which is not a very common way of zipping data. I think most of you are probably familiar with WinRAR or zip. However, for KeyGene, you need BG-zip, which is a typical way of storing the data. This is how a proper VCF file looks like, even more complicated than the files that I previously discussed. So there is a lot of metadata at the top that you where you see these dashes in front of, and this often tells something about how the way or how this data was generated, which reference was used, the version of the VCF file, as well as information about the tags that are being used in a VCF file. So the first column of a VCF file stands for the sequence identifier. We also saw that with the GFF file, it needs to be identical to the FASTA file where this VCF file aligns to. Then the second column is the position of the feature, and also in a VCF file, the positions are 1 based. So the first base of a genome in a VCF file starts with 1 and not with 0. The third column is a identifier. However, this can be optional. Then we have the fourth column, the reference allele, which is usually just GT, C, or A. However, in the case of a deletion, a reference allele may consist of multiple bases, which you see here. And then it means that at this position, so 1, 2, 3, 4, 5, 6, 7, the G starts at this position, the T and the C follow after that. Then you have a column for the alternative alleles. In the most simplistic case, you will only see 1 alternative allele in your VCF file. However, the more samples or lines or genomes you add, the more likely you will find different alleles, and then things can get complicated very quickly. So in this case, here we have a SNP observed on this position. However, we found a sample that does not show the reference allele, but only alternative alleles. And I will show you later how I can see that. But the list of alternative alleles can be endless, and it depends on how many lines you have and how complicated a particular region is. So especially in repetitive areas or in low structured areas such as small repetitive elements, you can find many different alternative alleles among samples. And these alternative alleles can also differ in size. So we have, for example, here the reference alleles GTC, which have been replaced by a G. but it also has been replaced by GT, C, and T. The next column is the quality, that says something about the reliability of a particular variant. There is a filter column that says whether variants passed filtering criteria. And then we have the information column, and the information column refers back to the metadata that is mentioned on top. So if it says here NS is 3, then here you can see what NS stands for, number of samples with data. DP stands for depth, AF stands for allele frequency. Those two are, I would say, always present in a VCF file. And it's even possible to make up your own identifiers as long as you mention them in the metadata. And KeyGene, as mentioned, expects a couple of fields to be present always. Then the format. The format means the way that a variant is built up. So in this case, it means that a variant is built up of the genotype, followed by the genotype quality, followed by the read depth, followed by the haplotype quality. You can find the explanation of these 4 identifiers again in the metadata. So if we look at this particular sample, NA0001, you will find that the genotype is 00, and 00 basically means I observed only reference alleles. The 0 stands for the reference allele. And it's often common to use this way of describing the genotype because the VCF file was, I assume, originally made in a diploid situation, and therefore it likes to pretend to have 2 alleles, but you just say allele 1 is 0 and allele 2 is 0, so I only observe the reference allele. Anything higher than 0 means alternative alleles, but we'll show you that. So the 48 stands for the genotype quality, 1 stands for the read depth, and 51 stands for the haplotype quality. Then going to this example, here we have a variant that says my alleles are 1 and 2. Well, the reference was 0, so for this variant, no reference allele was found, and the 1 refers to the first alternative allele, and the 2 refers to the second alternative allele. So this particular variant was heterozygous. I observed both the G and the T, and I found that in total in 21 reads, etc. Then you may also notice a difference between a piping symbol and a slash symbol. So a slash means a standalone variant, whereas the pipes refer to the fact that they are located on the same haplotype. That's more details you can read in the GTK specification, for example. So that's the way how VCF files are built up, and you can have multiple samples in the following columns, and the more samples you add, the more complex these 2 columns get. Then genetic maps, they are built on the concept of co-segregation, a common term with that, centimorgan positions. So 2 markers that are close to each other in terms of centimorgan positions tend to be inherited together more often. And by making recombination populations, you can make a pretty good genetic map of a genome, and a genetic map you can use in order to order contigs or scaffolds or any form of sequencing data to a chromosome level. The same accounts for QTLs, which also deals with co-segregation, but then linked to phenotypes. And physical maps can be compared to genetic maps. Physical maps use exact genomic coordinates, and genetic maps use estimations in the form of centimorgans. But basically, the order between a physical map and a genetic map can be compared, but also integrated. Physical maps and QTLs have start and end positions, whereas centimorgans obviously have not, because those are rough estimates. QTLs are often stored in the so-called map GFF format, whereas genetic maps are often stored in the DM1 format. So you see the map GFF on the left and the DM1 format on the right. We have some miscellaneous data types. Well, the gene passports were already covered. RNA-seq data is a very complicated type of data set, which I will not cover now. The same accounts for metabolomics, proteomics, and microRNA data. There are ways of putting this into KeyPedia, but it's for sure you need custom parsing of input data from the various analysis tools in order to make them KeyPedia compatible. These 4 data types are simply not standardized enough that every single data analysis package outputs the same format. So JSON knowledge is required in order to get that into the PDA. So if you want to have a look at how that then visually looks, so almost all data that has a visual aspect is stored in GFF3, BAM files, BigWig files, or VCF files. And I already mentioned FASTA at the beginning, but FASTA is not a visualization format. FASTA is the anchor point for these 4 data types. So we've seen this before, and hopefully you will now recognize the types of data that are behind the visualization. So all these data sets that you see right now are GFF files. All of them. They define the coordinates for specific features. And also here, again, GFF files. We've seen this already. Here we have some different types of data in there. So at the top, you see GFF files. So right here, but you also see some other visualization popping up. And these are other formats, can still be a GFF file, can also be a BET file. But by clicking on the track and looking at the information, you will exactly see what type of data this was. A BAM file in KeyPedia looks totally different than what we've seen so far. So at the top, you see again an example of a GFF file that describes the genetic properties of a gene. In this case, in pepper, Maor. And here you see a data of a BAM file. And the BAM file often contains read data. And if we do sequencing of genomes, then you have a usually tremendous amount of coverage. So that's why you see that these features go down quite a lot. And also here the max height has been reached. But BAM files are mainly used for an impression, not a very accurate single data point. BigWig files, as mentioned, display coverage. So you can translate the BAM file that you see here to a coverage representation. And that, of course, means a huge reduction in data loads in the PEDIA, because now all data points are just collapsed to numerical values. But depending on what you prefer, you may opt to visualize BAM files or BigQuery files or both. Doesn't really matter. It can serve as the same goal. And again, here gene passports, so GFF format. A VCF file in a genome browser looks like this. So just vertical bars with a color. We also saw the Variomics Viewer, which is a more extensive way of looking at variation, also with filtering options and comparison options. That's something that the genome browser does not have. If your VCF file is annotated, so that means that you need to have a genome, you need to have a genome annotation, and then you do a prediction of the variance that you see. What is the likely effect of this variant on the location where it happens? And the most common tool to do that is SNPF. And SNPF annotates your variants and assigns classifications to that. And if you're looking at variants in genes, causal variants for a particular trait, then you most often are interested in the variants that actually do something to a protein. And the darker the color, in this case red-orange, the more serious this potential variant may be. Low impact dark green, modifiers that do not end up in the difference for the protein are gray. And when there is no data at all, your bars will be blue. But this is optional, although very nice to have. The gene passports, we have been over that. You need custom-made JSON formats in order to get this picture working. And it's not data that can be obtained from the public domain. However, you can combine data from the public domain and make a KeyPedia compatible JSON of that, and then you have your gene passports. This was the genetic map viewer that we've already seen, so that requires the DM1 format. Often genetic maps can be found in public domain, usually limited in the amount of markers that are available. And if you have a very good proprietary genetic marker or genetic map, then you can of course add that to KeyPedia. This was the Variomics Viewer that has changed a bit in the way that it in the way that the side panel operates and looks. So the WeedPedia has the new version, although there was no data to visualize. So therefore, I simply show the old one. However, the concept in the middle remains the same. So you need a VCF file in order to visualize variants. You could do that in the genome browser, and you will see vertical bars. But in the Variomics Viewer, you will see this. And you also have a lot of options to narrow down this view. So the larger a particular area, and I mean, it's probably not visible for you, but here you see the different chromosomes, 1 to 12. So the darker an area, the more variants have been found in that block. And the Variomics Viewer can perfectly be used to search for patterns or to make contrast or to filter for high impact variation. Then what was already mentioned at the beginning, for the WeedPedia specifically, a download functionality was made available. So usually we will do that via the APIs. However, you need a a certain amount of knowledge to be able to operate with APIs. So that's communicating via URLs. So instead of doing that, we made a download page available, which is usually only available for the administrator. However, we also made one which is available for the regular user. And of course, that regular user can only download data sets that have been assigned to that user, so has the proper rights to access that data. So yeah, all the way at the bottom, you will then find a download link to download Weed data. Unfortunately, as mentioned at the beginning, you are not able to upload your own data. Again, you need to do that via an administrator, because you can imagine if all of you would upload your own data in different formats from different sources, that Wikipedia will soon be a complete mess. So that's why an administrator has the duty to properly maintain the whole database. So this in detail about data types. I can imagine that it's a lot, for some very relevant, for some of you probably not. But if there are any more questions on this, then I'm happy to answer them.
That's great. Thank you, Koen. Really nice overview of, I feel like the different file types and what they'll look like in there, especially when you're looking at the variation viewers, seeing the effects of different mutations within a gene. That's really cool, and for everyone to keep in mind that that's what we can work towards with data sets. So we will we'll have a designated administrator within the IWGC to upload these data sets. So as you go along, let's say you have a VCF file, result of mapping data that you want to put into the WeedPedia so that you can visualize it and work with it, then that'll request will come to our administrator, and we'll put that in, make sure it meets the formats and requirements, and would go in, and then you can see that and work with it within the database. So that, again, it's just exciting to think about the potential for this as we move ahead. So with that, are there any questions for Koen on how to, as far as the different types of data sets and what they'll look like, and anything along those lines?
Hi, Koen. I just have a question about the SNP-F annotated VCF files.
Mm-hmm.
So when you annotate a VCF file with SNP-F, obviously you get this big chunk of data in a new column just called info. Typically, I like to parse out pieces of that are helpful. If we were sending our annotated VCF to an administrator to upload, would we just want it in that raw, annotated, just big chunk of info?
Yeah, so personally, I would go for the raw data. The reason for that is metadata of SNPs are not displayed in Wikipedia automatically. So in a genome browser, you would only see the vertical lines, and if you want to see the annotated information, you have to click on it. And then it's just as user-friendly to display all the information that is there, opposed to an extract. On the other hand, SNP-F has a way of formatting data that, again, is not very user-friendly for humans. So I can perfectly imagine that you want to reparse this information and display it on a different way. So yeah, either are fine.
Okay, thank you.
Any other questions about data format or data file types? I think hopefully, again, a lot of that will be useful information for the afternoon training sessions when you'll be, for example, talking about how you map genetic data, so you know that's kind of the output of what you're working towards, and you'll work through the process of how to generate that, or same for RNA-seq, the types of files that are generated from that, or from population genomics analysis. So yeah, with that, I'm not seeing any further questions. So I've just a couple housekeeping announcements. The session, the afternoon training sessions, again, we have 3 of them. So they'll start at 1 p.m. and they run for an hour and 15 minutes, and they'll have you'll have a 15-minute break before the start of the next one. So they'll start at 1, 2:30, and then I believe that would be 4. The last one starts. So you see the room assignments over there and everything. For the participants virtually, for those, you're going to be in a Zoom room basically, so you can either use the chat box or your microphone to ask questions rather than through the Chime Q&A. And please, but feel free, you can use the forum during the day or the Q&A to ask questions to us. The kind of, if you have a general conference question, feel free to do that. I would like to say a big thank you to Koen and for the great overview of WeedPedia today. I really appreciate it. I think there's a lot to go through, so we're glad that we've been able to have this overview and introduction to the functionalities within WeedPedia. And we really look forward to the continued work with KeyGene over these next few years as we build out the database. So I think you might be able to hear us if we can give Koen a round of applause. So thanks so much. Yeah, have a nice evening there. And
you're welcome.
And for everyone else, we have lunch out just outside the conference room here being served. So enjoy the break, and we'll see you at the training sessions this afternoon. Thanks again, Koen.
Yo, bye-bye.
Good morning, everyone. How's the sound? A bit of a sound check. Yeah, you can hear it okay? All right, well, we're going to be starting in just a few minutes. We're working on the audio connection with our speaker in the Netherlands. Can I get a touch up, please? Good? Okay. Are we good to go ahead and get started, Will? Good to go. Okay, awesome. Well, this is so exciting, everyone. This morning is not quite our official kickoff. Tomorrow morning, we have some welcomes from our sponsors through NIFA and FR. But this morning, we get started. Today is our training day, and one of our 3 objectives for the International Weed Genomics Consortium is to have training and networking, and that's really what we're achieving here at the conference today. So welcome to Kansas City. For everyone who's here and to everyone virtually, welcome, and thank you for joining us. It's a great pleasure for me to introduce our training speaker for the morning. We're welcoming Koen Nijbroek. He is with KeyGene in the Netherlands, and KeyGene has developed an online database and platform for genomics called CropPedia, but in our case, we'll be calling it WeedPedia, of course, Weedomics. And this morning, Koen will be taking us through the functions of CropPedia, so you can understand how to search it, how to work with the different features for the genomes that will be uploaded over the course of now and the next couple of years. One thing I'd like to make sure everyone knows is that you can type in questions for Koen on Chime. So when you're in Chime, you'll see a function called Q&A, and that's in the lower left-hand corner. So please, at any time when you have a question, type that in. And then when we have the breaks, that's when we'll read those questions and get answers for you. So please don't wait until the end. You can type in questions during that Q&A time as well, of course, but there'll be just a little bit of a delay. So I encourage you to type in your questions as you think of them. And then for everyone in the room, we'll have the microphone for you so you can ask the questions as well for our speaker. So with that, again, it's a real pleasure to have you here. So excited to see everyone. And we will turn it over to Koen for our KeyGene training session. Thank you.
Good morning, Kansas. Nice to be present here. My name is Koen Nijbroek, born and raised in the Netherlands. Currently working for KeyGene, a company based in Wageningen. I will guide you through Croppedia, WeedPedia by presentation / workshop. So the way that this morning will look like is I will start with giving a general introduction about the core functionalities of Croppedia, then switch over to the pedia that you are probably most interested in, the WeedPedia. And since the WeedPedia is fairly new and still fairly empty with data, we will give a live demonstration by a rice use case. And then later on, I will also in detail explain about all the different types of data that you can put into Croppedia and how you can visualize that. So there will be breaks and Q&As in between, so don't hesitate to ask any questions. First about KeyGene. KeyGene was founded in 1989 by 4 Dutch seed companies. And even though they are all competitors to each other, they decided to combine their forces and create KeyGene to do a lot of science there. And we define ourselves as the go-to ag biotech company for higher crop yield and quality. We specialize in crop improvement by molecular breeding. And one of the tools that we made is Croppedia. And unfortunately, I only am present virtually. I was in Kansas City 2 years ago, but due to COVID, I was not able to attend physically. Nevertheless, this should be more than fine. So, since a lot of data has been generated proprietary as well as is present publicly, there is a need to combine all that data together and to interfere all the data for your genomic and genetic research. And that's where Croppedia comes in. And Croppedia is a single system that can combine pretty much every single type of genetic and genomic data. And then you can think of gene annotations, variomic data, RNA-seq data, proteomics, metabolomics, and it even includes PubMed literature as well. And this is how it looks like from the front page, and we will go into the pedia later on, but I think it makes more sense to explain the core functionalities first. It's a, again, single system where all the data is stored into, and you can interfere with the data by 6 main functionalities. And we will go through each and every single functionality in more detail later. But the 6 functionalities are Genome Browser, Genetic Map Viewer, Variomics Viewer, a data browser, a local BLAST tool, which has the same capabilities as the NCBI BLAST, but then the advantage is that you don't have to do it online. And it includes functionalities for omics analysis. So the home screen will basically guide you through each and every single one of these functionalities. On the right pane, on every single page, you can find an extensive menu where you can also browse through these functionalities, but where you can also find extensive help sections. So if anything is unclear at any given moment, you can just have a look at the help. There's an extensive search interface. So the more data you put into Corpedia, the more results you will get. And if you are working with a specific crop or a specific reference genome of a crop, you might want to enable or disable certain data sets. So with advanced searching, you can basically decide what you want to retrieve from the database. And as you see here, there's an example of a Solanum database. There are a couple of varieties in there, like Lycopersicum, Habochitis, Pinelli, Arcanum. If you're only working on Solanum Lycopersicum and you're only working on the latest, well, it's actually not the latest genome, but version 3.0, then you can just enable 3.0 and disable all the other databases. Once you hit a search term in Corpedia, you will basically see a window that will come up with all the results that have been found of all the data that is in the system. And that can be a lot. So if you type in a generic search term as flowering or yield, every single data component in the system that contains that exact word will be shown back. And we will go into the details of this search later. But on the left top pane, you will see gene passports. And basically, that's the same as a passport that each and every one of us has for traveling. It contains all the essential information about a specific gene. We also have a pane for genomic features, overviews, genetic features. You will see a pane where on a genome all your results are located. So if you are doing some specific research, you might end up that all your search terms are roughly located at the same genomic locus. There is a link with PubMed where you can browse for literature. And at the bottom pane, we also have pathway information whenever relevant. One thing in Corpedia that you can do is make a digital notebook or lab journal. So every single data entry that you find in Corpedia and that you might find interesting for your scientific research, you can save it in a workspace. And basically you can give it the layout and structure that you desire. You can even share it with other colleagues or users or generate extracts out of that for other purposes. So the main core functionality of Corpedia is the genome browser. And I hope that most of you are familiar with the concept genome browsers. It's widely used in the public domain as well. And basically what it does, a genome browser is a representation, a linear representation of a genome. And you can visualize all kinds of properties of that genome. A genome browser is limited to only be able to view one genome at once. So if you are interested in multiple genomes, as we see on the screen here, you have to open multiple genome browsers. So this is what it looks like. This is obviously the genome browser tailored to Corpedia, but it resembles quite well with common genome browsers in the public domain. So on the top pane, you will find navigation features that will allow you to go to certain positions or regions in your genome, to zoom in or zoom out the region that you are looking at. And on the left side pane, you can enable what we call tracks. So every data file that goes into KeyGene is called a track. You can enable or disable that track to see the data that is in there. If we zoom in a little bit and only look at a single gene, for example, you can see the whole genomic region. So you see that it obviously starts with HEG. You see the different reading frames that are possible. You see the protein sequences that are possible. And you can then look into details at features. So if you would select this particular gene, which was done on the right bottom, then you can see that you have multiple actions that you can perform on this specific data feature. You can save it to that digital notebook that I just mentioned. You can download sequence. You can search for it in KeyGene to find other relevant hits. You can pull up that passport, which contains all the essential information about the gene. And you can download also kinds of other data types. So going into the gene passports, as mentioned, it contains all the essential information that a gene might contain. And this is not data that can be easily obtained from the public domain. So data that you download from the public domain is usually limited to a certain aspect, whereas gene passports try to combine all different kinds of aspects. So you need to either run a sophisticated gene annotation pipeline or try to combine all kinds of data together. But if you have that data, what you can do is look at accession information, look at predicted localization, interpro results, BLAST annotations, GO accessions, transcription factor information, phylogenetic trees, protein clustering. But we will go into this in detail more later. So this is the search bar again, and also from there you can browse to the gene passports. Also from the genome browser, you can browse to the gene passports. So all the data in KeyGene and all different ways of approaching the data is also linked to each other, so you can easily navigate to specific features. In KeyGene, you can compare genetic maps. So if there are genetic maps available of the crop that you're interested in or the genome that you're interested in, usually you will see that genetic maps from the public domain are relatively limited in the number of markers that are present. But if there are a variety of different genetic maps, you can easily compare them and see whether markers are in the same orientation or have the same physical position. So that's what you see here, where we enabled a genetic map and linked it to another genetic map, and then linked it to another genetic map, and so on. And you can see here all the connections where similarity between the three genetic maps were observed. Here's a closer look into that. So you see, for example, here that there's one match between these two genetic maps, but then none after that, whereas another genetic map, which is not observed here, contains far more, many more matches. KeyGene also has a BLAST functionality. So it's KeyGene is mainly intended for visualization and data interrogation, not necessarily data creation. So running very computationally intensive blast jobs, Oncopedia is not recommended. For that, you probably would ask a bioinformatician to do a blast for you and then visualize the result of that. But nevertheless, you may have some proprietary sequence information that you do not want to put into the NCBI database, or maybe it's not the policy to allow for that. So for simplistic blast jobs, you can do it in Incopedia locally, so your proprietary information is therefore protected. It contains all the same functionalities as what you would do with the NCBI blast. So you can choose for the different types of algorithms. You can go into the advanced settings and change values whenever applicable. So the results of that will be only visible for you once you are logged in. You can again save it to your digital notebook, display it in a genome browser, and from there you can also again share with other colleagues. And here you see an example blast job, and hopefully you're all familiar with the output of blast. If not, we will go into detail in the last section of this demonstration. But here you can see that we found a match on the Solanum lycopersicum genome chromosome 7. Another functionality in Incopedia is the Variomics Viewer. So if you have a lot of resequenced lines or any other type of resequencing data that's often stored in a variant calling format, VCF. Also that we will discuss in detail later, but there is a specific functionality for that that can load VCFs and display them, and from there you can very easily search for patterns or functional SNPs. You can also display it in a genome browser, but the Variomics Viewer is a bit more detailed and extended for displaying variant information. So this is how it looks like. On top, it's the same linear representation as a genome browser. And then on the vertical axis, you see the different accessions that are in there. And the more intense the color of a specific block is, the more variation has been observed. And you can already, this is just a view of all the variation of all 12 chromosomes of, in this case, lycopersicum. You can already see that there are a couple of accessions that have the same SNP patterns, whereas there are other accessions that do not have this specific pattern. So if you're interested in, for example, a group of lines that are resistant to something or show a specific phenotype versus a group of lines that do not, then you can easily make these visualizations and search for patterns or SNP annotations. So there is a extensive way of narrowing down that whole view of the genome and to search for variation of interest. You can make contrasts, you can apply filters to how variation may behave over a certain amount of lines. There is, if available, SNP annotation, and then you can filter on the impact of variation. You can also filter on the quality and the depth for retrieving variants that are more reliable, for example. So that's the core functionalities of Incopedia. So now we will go to the actual WeedPedia, have a look and feel of how Incopedia works and operates, and see what is already available for you. So this is the WeedPedia website, weedpedia.weedgenomics.org. and I will for now use a KeyGene login. But if you have a generic account, then you are able to log in and view all the data that is available for that generic account or for you. So there is a lot of proprietary information in this WeedPedia as well, which is specific for certain individuals, and you will not be able to view or extract any of that data. You can only see the data which has been assigned to you. And as a general user, you will be able to find two different types of data sets in the WeedPedia. Passia scoparia, which is, of course, a type of weed, and the other one, and I think the common name is horseweed. There are two genomes available in the WeedPedia for all. And there is, at least for one of the genomes, already some gene annotation available. But we will see that later. So now this should look familiar. We have these 5 core functionalities: genome browser, the map viewer, the Variomics Viewer, BLAST, as well as omics analysis. You can make workspaces, and we will do that later with the RISE demonstration. And on the top hamburger menu, you have different options. So we have these 5 core functionalities again, but you can also opt for the help. And if you click the help, you will find an extensive section that will tell you everything about the most important window of Copedia, the homepage. And it will explain everything in detail much more than I will do now. So if we have a look at the weed data that is already available, if we have a look at the first genome, then you will see 2 different tracks available in the WeedPedia. So the first is the reference sequence, and the other one is a genomic feature, which contains gene annotation. So on the top pane, you can browse through the genome of this weed and have a look at the regions or genes that you are interested in. You can either browse or navigate to areas, but you can also type it in. And if you have very specific search terms or names of genes, then you can also just enter it in the search and then go to it directly. You can zoom out and zoom in, obviously. However, the more you zoom out, the more data will be visualized, and you can already see here that the max height has been reached for this specific track. So there's a lot of data available still here, but you're not able to see it because you're now looking at 1 MB of sequencing content. So that's a lot. There are multiple ways of tailoring your visualization. So every single track has this small button next to it, and then you can edit the configuration or the way of displaying. So if I would opt to collapse this track, then everything will be condensed in one single entry. But then you also lose a lot of data that you would otherwise see on the screen. So it depends on how you want to visualize your data and what you want to see. But in a normal situation, looking at a very large area, it's simply too much to display. So there is a way, of course, to zoom in, either via the top panel or by just using your mouse. And we can have a look at one of the genes that is available. So if I would do a left click on this, then I would be able to find some metadata of this gene, where it is located, the size of this gene, a description, attributes, the name of the data set that it came from. And I'm also able to extract the sequence. So starting from ATG all the way to TAG. You can either copy paste it from here or you can download the FASTA directly. Bear in mind that this is the genomic area. And if you only want the coding region, then you have to scroll down a bit to the mRNA, which is a bit shorter than the sequence of the whole genomic region. You can also have a look at individual exons. You can download individual exons, etc., etc. So there's already a lot of information by just clicking on the feature. If you do a right click on the feature, then you have a lot more functionalities that you can use. So by view data details, we will see the screen that I just showed now. But there's also the possibility to highlight, for example, this gene. So if you're working in a very large window and you want to focus on a specific area, you can also just highlight it. You can add it to the workspace. You can copy the sequence to your clipboard. You can download the sequence, send it to BLAST, use it for search, and also have a look at the gene passports. So in this case, gene passports have been made for, I think, both the genomes that are available for all of you. So there has been some extensive gene annotation going on. So if we look at this specific example, we can see that it is a nuclear transcription factor. It's located here. This is the taxonomy ID of this specific weed. We have the coding sequence here. We have the protein sequence here. The predicted localization. And if you're unfamiliar with what that means, there is a small description that you can hover over. So, well, it's pretty obvious in this case that the predicted localization is nuclear and not necessarily cytoplasmic. We have Interpro results, which is a public database, and it also contains links to Interpro. So you can easily go to the public databases if needed. We have BLAST annotations as well. So there's BLAST information in here. Go accessions from the Go database, and also here it describes that it is likely to be present in the nucleus, has something to do with transcription, etc., etc. So you have different types of data sources that will tell you what this specific gene/protein might do and where it operates. There's even some RNA-seq data available for this specific gene, but this is just test data to play around with the functionalities. And if we would have Variomics data available for this specific genome, then we would also see a box with variant information here for just this gene. And that allows you to easily look whether you find interesting SNPs or variation for certain lines. So if we go back to the main window and have a look at the comparative map viewer, well, there is no genetic map information available for this specific genome. Doesn't necessarily mean that it's not there. So, I mean, if you find relevant information from the public domain, you can always ask the administrators to make that data available in KeyGene as well. So bear in mind that a regular user cannot add anything to the system. It can only visualize data and interrogate the data, but making data available can only be done by an administrator. We have the Variomics Viewer here, and this looks different than what I just showed in the presentation, and that's because the WeedPedia already works with the new version of the Variomics Viewer. However, there's no data yet available, so it's also not something that we can have a look at now, but it harbors the same functionalities, although the visualization has changed somewhat. If we go to BLAST, well, you can, every single user can do a BLAST in KeyGene, and it's only available for that specific user. So you can either type in a sequence or paste a sequence in here, define what type of BLAST you want to do. You can check some of the BLAST options, and you can also define to what database you want to do your BLAST. But also here, there's no publicly available BLAST information yet. The last part is the omics analysis, and I did not share that in my presentation, and that's because this is quite new in terms of functionalities. And recently there has been some test data put into the system. However, this test data is still fairly limited. So at the end of section 3, when there's still time left, I will show a video about all the RNA-seq features and functionalities, because it's a lot, and by going through the test data, you won't get all the essential information that you might want to know. Lastly, you can make a workspace in the WeedPedia. Again, it's just for you. However, later on, you can share the workspace with other people that have an account in the Pedia. So by just creating this workspace, I can store all the scientific research that I will be doing. So you can edit text, you can paste pictures in there, and all the data types or all the data in KeyGene that you might find interesting, you can store it to this workspace, and it will be visualized here. And later on with the RISE demonstration, we will have a closer look into the workspace. So there was one other genome available already, publicly available, horseweed, and also here there are only gene passports available. Well, not only, I mean, gene passports contain a lot of information, but there's no variomic data yet. There's no genetic map information yet. But I'm pretty positive that that will still be coming or will be generated later on. So this will hopefully provide you some insight of what's already available in the WeedPedia. If you have any questions on the general concept of KeyGene, feel free to ask them. If you have any questions about the data of the WeedPedia, unfortunately, I'm not the person that can answer your questions, but you probably know who can. So I see that went by very quickly. So already, 20 minutes ahead of schedule. So that means that we have quite some time for questions. So yeah, go ahead.
Right, so if anyone in the room has a question, we'll come around with a microphone and you can ask your question as well. We're watching online for any questions to come through. And as you were seeing, we've been working on with KeyGene to upload these genomes that are publicly available so far to get them in. And Eric Patterson and Nathan Hall at Michigan State have been working really hard to develop these gene passports, what you're seeing. So there's an incredible amount of information that's already been searched and compiled from different databases that comes up at in one click when you're looking at any single gene. So that process, they've been spending several months to get that together, get it working, and with the KeyGene team to have that all integrated. But now that that's working, as you can see for kochia and for horseweed, then now there are a number of additional public genomes that we'll bring into that database, as well as the other genomes as they get finished. So that part is really exciting. And but yeah, on this first part, there's kind of the BLAST functionality, the gene passports. Or if there's anything there, if you think there's some information you'd like to have when you look at a gene page that wasn't there, have suggestions for what to add. All right. Yeah, so sorry, we do need, I'm wondering, Carlos, could you help us out? If you could go to the back and grab a microphone, please. And then if you can run it around to the people asking questions. Thank you. Appreciate that. So Koen will just have a microphone here in the room, and then they'll go to the person asking the question so that you can hear it.
Sure.
Hey, does it work?
Yep.
Okay. So is it only going to be public genomes, or is there sort of like an incentive for people to send you genomes that they are working on?
Yeah, so I mean, WeedPedia is suitable for public genomes and proprietary genomes. So the administrator of the WeedPedia basically has the capabilities to assign data access. So whenever one of the partners has a specific genome that they want to add into the system, but should only be visible for them or maybe a certain group of people, then the administrator can make sure that that data only is visible for the persons that have the proper rights to see that data. So there is already a lot of proprietary data in there, but of course I can't show that to the general public.
Thank you.
I would add to that we do have a list of genomes that we're planning to put in the WeedPedia, and Sarah Morin's talk tomorrow will go through the list of, there are a number of published genomes that are out there now that are good enough quality to put into the database. And then the consortium is working with a number of people who have projects in progress to maybe help out a little bit to get it finished to a certain level and then post it. There are various groups that you'll see who are working on genomes independently, and they've agreed once it's finished they'll make it available so we can put it in here. And then this the ability to log into WeedPedia will be available to anyone who wants to be part of the consortium. So just basically, do you have an interest in Weed Genomics? We'll set you up with an account. It's free to have the access. And then as Koen was saying, we are doing 10 genomes through the consortium. And again, in Sarah's talk tomorrow, you'll hear more about those. But the founding members, who are the sponsors of the consortium, have access to those for 1 year first. So the data administrator will designate that they can see those for that first year, and then once that year is up, then they'll be available for everyone to see. So, and we also have a question online from Tan with BASF, what other weed species will be in WeedPedia soon. So I'd say Tan, yeah, we'll cover that in detail tomorrow, but we are anticipating that by the end of, by about 18 months from now, we'll have 34 weed species in the WeedPedia, which is pretty exciting when you think that, you know, the Coniza canadensis genome was published in about March 2020. So we're gonna have a nice inflection curve here in the number of genomes coming into the database. But another part of the consortium is to give the ability for people to just communicate, research networks, update on what's going on, because there are certainly people out there who are working on a weed genome that maybe the consortium maybe doesn't know about, and we want to work together, or you want to host it in here. That's definitely an option. All right, any other questions in the room on this first part? Koen, I have a question on the data management. So if we go to the hamburger menu, that's where for the genomes to which a user has access, they can actually download the FASTA file, right, if they want to download that genome themselves?
Yep. So yeah, that was a sort of spoiler because that was intended at the end of the last section, but it doesn't matter. I mean, we can also do it now. So yeah, all the information that a user has access to can also be downloaded. So I can imagine that when you get to work with the WeedPedia, that certain data sets that were already in there, that you might not have these available locally. So in the hamburger menu, there's indeed a button, data management. You will see again these 2 genomes that will be later on a very extensive list. And you can have a detailed look to what types of data is present for downloading. So here, for example, it says that there are over 612,000 mapped features that you can download. So that's the genome annotation that's present there. There are BLAST databases that you can download. So, I mean, you don't even have to make your own BLAST database if you want to do stuff locally. There's RNA-seq information in here, and also for the other genome, again, genome annotation. So if you would enable one of these features, you will find some metadata about this data set. So in this case, I selected the mapped features, and it says horse wheat, number of files, taxonomy ID, what type of file it is. So it's a GFF, and the source where it came from. So if I would now click on this GFF, you can already see at the bottom of my screen that you will be able to download the file. I'm not going to do that as an example, but believe me that it works. So that's the way of downloading information from Corpedia and use it locally if necessary. Something else that we that you can also do, there is an extensive API present in Corpedia, and not everyone will be familiar with APIs or how to use them. But basically, by making use of URLs, you can download files or retrieve data from the system without you having to browse to it. So if you are interested in using the API, and there is a very extensive manual available that basically shows you how to access data via URLs and what the output of that is. And you need access tokens for that, but by then the Corpedia administrator can of course provide that for you. So there's really a lot of functionality available to basically do everything in Copedia, but then by navigating through URLs. Does that answer your question?
Yes, thank you. Do we have any other questions here in the room or online? Again, you can use the Q&A link that's in the lower left-hand corner of Chime, and we can read those questions out. All right, well, I'm not seeing any more questions here in the room or online. So do we want to maybe take just a little bit longer break then?
Yeah, that sounds good. I mean,
Okay.
there is a possibility to start the RISE demonstration earlier, but yeah, if people were waiting for that and they tune in at 9:45 or at 10, then obviously they miss a bit.
Yeah, how about let's do that. Let's plan to we'll come back here as scheduled at 9:45, right?
Mm-hmm.
I think that's when session 2 starts.
No, it starts at 10.
Oh, it starts at 10.
Yeah.
Okay. Yeah, but we probably don't want to if people are following the schedule online, they'll be that. Well, that'll give us a little time here to have the coffee break. And is it possible? Oh, yeah, go ahead. Okay. Well, I think that's probably what we want to do. So the next session is scheduled to start at 10, and then and that's going to be covering. You go into more detail on I forget which sections are we covering for that part.
Yeah. So the next session will be a demonstration of Copedia with a use case of RISE. So there we have a very precise scientific question that we want to address, and then we will see how Copedia can answer that question.
Okay, excellent. Well, that does make me think of maybe one question, and just to show everyone some of the custom search features that we've put into our gene passports. Could you show them, if you could share your screen in WeedPedia with the Bassius-Goparia genome, and show if you type, for example, EPSPS into the search term to look specifically.
I will do it from here.
This is one of our genes that people tend to look at quite a bit.
Let me see if we have. Yeah, we have it enabled. Okay. So here's the search result for EPSPS.
Yeah, and then maybe if you click on the gene passport, and you can see.
Yeah, so then we end up indeed at the window that we already had a look at before.
Yeah.
And indeed from there you can again go to the genome browser, add to the workspace, etc., and have a look at what this apparently very important gene does.
And if you could go back up to the top, they've Nathan and Eric put in some custom terms just so everyone knows. So you can see HRAC:9. So that's of course the herbicide group for glyphosate. So wherever if we have a known target site gene of a given mode of action, you can type in HRAC: and then that mode of action number. And if we've been able to annotate that target site gene in there, then you'll be able to find it. So that will work for ones where we know, if we have a one or a few target site genes. You can see also glyphosate, EPSPS. So some of the common kind of abbreviations, terms that people in our field would use. So this would work for ALS, HRAC2, for example. So there we don't have we certainly don't have every single herbicide, but I think at least as far as mode of action group, that can help you navigate to find, and that's in the pipeline to annotate that for every genome that's coming through. So that's pretty exciting. And they're also working on a pipeline to annotate anything that would be, for example, a cytochrome P450. So you can just type in P450 and you should get a list of everything that is predicted to have that function. Of course, with those, you have to do a lot more manual curation. The same for GSTs. So really, we're trying to build into the annotation some of the typical things that people would be looking about when thinking about herbicide sites of action. Yeah, I've got another question here.
Sorry, I was gonna wait until after the next section, so I may be jumping the gun a little bit here, but the herbicide target gene thing made me think. So if you upload a genome that's not annotated, can you compare across genomes and try to align and find these genes that are annotated in another genome?
Mm-hmm.
Yeah, it's an excellent question. The answer is yes. However, you need a bit more than just Cropedia. So to recap first, Cropedia is not a data generation tool. It's a data visualization / interrogation tool. So if you would have a blank genome in there, then you will need bioinformatics capabilities to make, for example, a synteny analysis between these genomes, or by mapping features of a particular genome to another genome. And then these data sets that basically contain the links between two genomes or multiple genomes, those can be uploaded into Cropedia, and those can then be visualized, and then you can exactly see that if you have genomic annotation from genome A and you then add a track with mapped data from genome B, then you can see exactly whether genes are present there, yes or no, or whether there are base differences or structural variants. So yes, it's possible in Cropedia, but you need to do the analysis up front.
Okay, thanks.
And later on in the third section, we will go into extensive detail on what types of data can be put into the system, so that you know how to generate the proper data files for Cropedia. But basically, Cropedia can handle every common format. However, as soon as you deviate from that format, then Cropedia will not accept your input data.
All right, any other questions? I'm not seeing any more questions online still. Well, feel free to take a minute to, during the break, think about the Cropedia, and in the next session, starting at 10 a.m. here Central Time, we will get more into some of the basic and intermediate functionalities of WeedPedia. And as Koen said, we'll look at the rice genomics data. So then you can really see, this kind of helps everyone think ahead. What can you do? What kind of data can you generate that can then go into the WeedPedia, and what can you do with it? So thank you very much, Koen. So yeah, we'll just have about a 30-minute break here then, and come back at 10 a.m. local time. All right, thanks everyone.